So we're 9 months into this COVID-19 pandemic. So many people have suffered so greatly, but one of the silver linings is that people have learned a lot more about science, especially the very complicated network that is the immune system. We're going to choose one of its fighters, a special type of protein called antibodies, and dive all in. Antibodies are one of the soldiers of the immune system. They work to snipe the bad guys invading your body. It's a lock and key relationship. Find the best antibody key to unlock the bad guy and defeat it. So how does your body make the best antibody snipers? That's where biological combinatorics comes in. B cells are where antibodies come from. The two parts of your immune system: innate, the built in general defense, and adaptive, what your body combinatorically learns to build, specific to invaders. We see the Fab binding to the invaders and the FC to get more of the immune system in the fight. The tips of the antibody, its variable regions, and the rest, its constant regions. All the pieces interact to allow and stabilize the best possible binding to the invader. Antibodies can hang out on the surface of B cells, which is why they're also called B cell receptors. Variable regions are the magic of antibodies. They're highly specific to their target because that's what makes them so neutralizing. That means the body needs to be prepared, have a fighter or a variable region for every possible invader through a nearly random shuffling of genes called BDJ recombination. It creates a sample space of antibodies that's very broad, so we cover as many threats as possible. That means that you usually have at least one antibody that you can get to fit as a key in the lock of the bad guy, known technically as an antigen. This happens at the epitope of an antigen. An antibody using its paratope of the Fab can bind to an antigen surface. So you're picking an antibody which best matches an epitope like a puzzle so you can shut down the bad guy and prevent it from wreaking havoc. Then, through something called somatic hypermutation or SHM, the body makes lots of random small changes in that best initial match so you can whittle down that puzzle piece to fit even better. Which is why, say, from a starting antibody, you get antibody 1 and 2 from SHM. But 2 may fit better because it's slightly more specific for where it attaches, so the body will try to make more of those. So if you're exposed again to the bad guy with that antigen, you're super ready to go. Science today can make antibodies in the lab to be medicine. Many groups have done this for COVID. We gather biological samples to figure out what the body does for natural infection. We improve and standardize, and we produce it as a drug to treat sick people. Such a drug could work in one or two ways to prevent the virus from getting into the cell, but overall, we're matching epitopes to make sure we have the best puzzle piece possible. We can also use antibodies to fight cancer and a bunch of other diseases. They're actually not just for foreign invaders. Sometimes with conditions like type 1 diabetes, lupus, or allergies, our antibodies attack something they shouldn't as a bad guy. But antibodies are still awesome. The COVID vaccine is so effective because it does a great job of safely playing the game of biological combinatorics, so your body finds and improves the best antibodies to beat the virus without getting sick. So if you do encounter the virus, you can fight it off without that whole process to make the best antibody puzzle piece. But the next time you hear about antibodies, think of this entire world of immunology. It's a complicated network, but if you pull at one thread, you can begin to understand the whole beautiful system.